Silent mode. ಸೈಡ್ಗೆ ಕೆಳಗೆ. Okay, am I audible to you all? Just confirm me once if I'm audible to everyone. My audio and video both are clear. Okay, so fine. You people were asking like when the plant science classes will start, when the English medium classes will start for the 2026 and 2027 course. So finally, we are starting the English medium classes for the plant sciences. And if you already know, the English medium classes for the plant pathology is already started, but I know like you were waiting to start from the genetics. So finally, I'm here and we will be starting the genetics classes. And if you want to know the schedule, so a lot of students were asking about the schedule first. Sir, what will be the class timing and what will be the class date and what will be the topics that will be dealt? We want the exact timetable. So the exact timetable, you can find it in our website. So I'll just show you like where you can find this thing, and you can keep yourself updated and you can attend the classes accordingly. Okay. I'll just show you once. So just if you open Google Chrome and search for AgriAddict, so if you search for AgriAddict, so if you open this website agriaddict.in, so here while closing this, just you have to click on this close button. This will be closed, and this is our home page if you know. And here you can find the course timetable. Along with the plant sciences, for all other subjects, the timetable will be available in this section. Just click on the course timetable. So once you click on it, you can just scroll down, and here you can see that the complete subjects are mentioned here, and it is also mentioned for like ICAR PG, CUT PG, SRF, and IBPS. Currently, the ICAR PG PhD classes are going on, so that you can find the timetable for all of this. So for now, the plant sciences English classes, if I click on view all, so here if you scroll, first thing what you need about like what topics will be dealt and when it will be dealt. So my classes will be regularly by the 10 p.m. So every day by 10 p.m. I'll be taking all the classes. So we are starting today with this topic of history of genetics and cytogenetics, and the date is mentioned here, that is 18th of November. Okay. So similarly, like I have given the schedule till the end of November. So for every month, the monthly schedule will be given. The timetable will be of only a single month. So after that, next for the December, the things will be updated here. So whatever blank you are seeing here, that will be automatically updated. You need not to ask us on the WhatsApp, you need not to call us, or you need not to be tensed about like when the classes will be, what will be the schedule, what will be the next class. But anyhow, if you all know like already that recorded classes are available of the previous batch, plus all this live classes will be also available as a recorded form. But I would definitely suggest you people, like if you are very, very serious about your academics and if you want to secure a good rank, the top rank in this ICAR PG exam of plant sciences, try to attend all the live classes, interact with me, be interactive, and just drop all your doubts, get it clarified. Anyhow, this is just like a demo classes which we are going to have it in the YouTube for the new students who do not know about our platform and who want to just check like how do we like teach. So after 1 or 2 classes, all the remaining classes will be in our platform, that is in our mobile application or the website. So you can directly take the classes from there. And once we start the classes in the website or the application, you can freely interact by unmuting yourself and ask all your doubts and get it clarified. And those sessions will be really engaging and entertaining. So that's what I can say you for all. Sir, what about SRF 2027? I want a 2 years course. So currently, like we have only launched the 21-year course for the SRF, because for the SRF we do not have that structure to launch the 2-year course. But one good thing is like one benefit if you are taking the 1 year course is also that, so you will be getting the extra 30% off in the next year. For example, if you are taking the 2026 course of SRF, right, currently, but once the validity is expired, for the next year you will get flat 30% off since you will be the existing student. So that we have given the provision for all the students. Okay, so you can avail it. So for the SRF, let me make it clear, we only have the 1 year course right now based on our capacity. So but if you want to take it for 2 years, just register now, but for next year you can avail up to 30% off on the next course. Okay, so now let's not let's not discuss anything about courses or something. Let's stick to the subject and let's start the discussion of the plant sciences genetics classes. Okay, so if you have any queries related to the courses or anything, you can directly contact on this number. So you can contact on this number and our team will help you with everything. Okay, but now let's focus on the subject. I hope you people are excited, and I'm very much excited to start these classes on the smart board. And if you know, like, this is the very first time that we are having all the classes on the smart board, and you people are, your batch, like 2026 batch, is very, very lucky that all the classes will be on the smart board and you will get the best experience possible, best learning experience possible from our end. Okay, fine. So before I start, I'll just explain few things. Before we start to the introduction to genetics, let me introduce myself. So I am Ari Prasad, SK, a student of IR in New Delhi, pursuing my PhD 4th year from the Division of Seed Science and Technology, and I have cracked ICAR PG in the year 2020 with 48 rank, ICAR PhD in the year 2022 in the All India 2nd rank. After that, I have also, in between, I have cracked this IPS A4 mains, and after cracking this SRF, I also cracked the ASRB NET exam in the subject of Seed Science and Technology, and I have a prior teaching experience more than 4 years in this background of genetics and seed science. Plus, I also teach crop physiology and the biotechnology, few bits, and also the biochemistry. So this is my expertise, and this is what I do. I'm doing from last 4 years, if you know it. Okay, fine. So next, let's start. So first thing, the master plan. So what is our plan? So if you are preparing for 2026 or 2027, just a minute, give me a minute. Sorry. So if you are preparing for the 2026 or 2027 course, okay, then I'll explain about the what plan we are going to have it for you. So most of the students are concerned about, like, sir, when the classes will start and when you're going to cover the syllabus and when we will get the time for the revision and whether we have to prepare the notes or not. How should we, like, cover the syllabus? So let me explain the subject of genetics. What we are going to do is, like, who is going to teach? Like, I'll take the complete genetics for the English, and for plant breeding we have Ari Vijaya. Like, if you have already seen the previous year classes from the YouTube, from the classes, Ari Vijaya will engage the classes, Rupa will engage the classes, and Abhishek will engage the classes, who has taken the revision sessions in the last year, and all of them are from IRA who are pursuing their PhD from the genetics and plant breeding. And next, if you know about the plant pathology, already the classes have been started by Parul, and we will also include 1 or 2 mentors, like the fresh students, like who have topped these exams in the 2025, along with the Parul for these classes. And for microbiology, the Ujjwal will be dealing with all the microbiology classes. He is a on-roll PhD student from the microbiology department, so that whatever content that he explains or he will give the classes, that will be lot useful, because the microbiology and seed tech is like a minor subjects. Every time they are neglected, not thoroughly prepared, and you if I ask you, like, what standard books do you refer for the microbiology, you will only say one book, that is Tara and Kapoor. Other than that, you do not know anything outside that book, right? But because, like, we are including the IRS students, like, they will be teaching all the updated things, so that mostly the questions that comes in the exams, we will be able to cover it. Okay, so this is about microbiology and seed technology. If you know it already, like, I'll be engaging all the seed tech classes, all the updated things, updated material, you will be getting it from my end. Okay, so this is about English. And about Hindi, the Paras will be dealing with Hindi classes. And then about the plant breeding, we have Mausam, who is like All India previous first ranker from the ICAR PhD exam, who is just joining the IRA for the PhD in the genetics and plant breeding. She will be going to deal it, and Vivek. And also along with this, we have like one uh, alumni of IRI who will be taking all the remaining classes, the advanced topic of this genetics and plant breeding. And next, if I talk about the plant pathology, Parul will be mainly handling all the classes of Hindi. Okay. And next for the microbiology also, Parul plus me, I will be trying to engage the Hindi classes. I will be supporting her in this part. Okay. And for the seeds and technology, hopefully, like I already explained that for seed tech, I don't want others to engage any classes because I will not be personally satisfied. So I'll try to engage us classes in the English language, but I'll try to keep it simple so that you will be able to understand each and everything in detail. Okay. Don't repeatedly ask same things. That's why I explained one time in detail. So if your friends, any of your friends in the master group or any of your friends to you, if they're asking like who is going to deal and how they are going to cover it, just share this video link. Okay. So what is the mini? So everything apart from the class, I will be only answering if you drop on the comment section after ending this live. Okay. So let's stick to the class now. So book list. So let's start with the genetics. So before I start, I'll also clear the book list so that there would be no confusion. I guess like some student asked me in this master group, some students also messaged me in the YouTube and also in the free groups like, sir, what are the exact books that we have to cover for the ICAR PG exam? So you already know about the BDC, but along with the BDC, if there are any other books necessary or needed or any complementary books that you can or supplementary books that I can suggest. So I will tell you like all the books that I will be referring to teach you people. So this gives you the complete idea. Okay. So if I'm right now telling you all the books that I have studied to teach you people, that would be of lot of use for you, right? So I'll tell you. So first thing was always like BDC. And if you ask me like, sir, whether you have studied like fundamentals of BDC book, fundamentals of genetics or just genetics. So if you ask me whether I studied the genetics by BDC or fundamentals of genetics by BDC, I have studied fundamentals of genetics, plus I have also studied the genetics. So if you ask me like, so what is the difference between these two books? So there is one name with a title called as Fundamentals of Genetics. There is one name with a title called as Genetics by BDC. So what is the difference between this? See, the Genetics by BDC is in bit detail. Okay, remember it is in bit detail. Whether you need it or not, that I cannot explain in one word. Okay, few things are required to get a clarity about the concepts. Few things are extra. So overall, it is like bit extra detail. And this fundamentals of genetics, if you have Xerox, if you have things already in the new edition, there is only one book. But in the previous editions, in the old books, there are two books: the fundamentals of genetics and genetics. So if you have this fundamentals of genetics, it is bit concise. It is bit detail, and above one is bit concise, so that limited information you will get. Not much difference. Assume that there is like 20 to 25%, maximum 15 to 20% of difference in between these books. But when I prepared for my ICAR PG exam, I only referred this fundamental of genetics. So if you are starting from scratch, you do not have any idea regarding like how to start and your foundation is not that strong, best thing is like attend my classes. But if you are not attending my classes and if you are not taking the master course, go for the fundamentals of genetics. So that is where you can start this. Okay, fine. So next, this one is clear. The second book which I've referred from last 3 to 4 years to teach you all is like Pathfinder book. So from this, like most of the molecular concepts, like molecular genetics concepts are clear. So that's what I referred. And the Pathfinder book. So this book, if you do not know, this is mainly for CSIR NET exam. Okay. So you will be knowing about these exams and all like after entering to the MSc. So do not worry about it. But if you want to explore, if you have find time, you can explore this, but not compulsory. Please do not take this as compulsory. Only compulsory book is first one. If you read the first book, it's more than sufficient. But still, if you have that hunger that you want to read multiple books, if you have sufficient time, then you can go for the other books. Anyhow, like if you try to attend my classes, all my classes, the total number of classes that I take in the genetics is like around 60 classes. Last time it was 55, but now this time I will take 60 classes. So if you watch all the 60 classes, which will be around a duration of 1.5 to maximum of 2 hours. Plus, you just study everything from the notes that I have already uploaded. I can frankly tell you that 90% of the genetic syllabus will be done. It will not just be cleared, you will be perfect for the 90% of the questions that are being asked in the exam. Sometimes it can also go to 100%, but I won't take that extra thing. I'll stick it to like 90% will be totally done if you just attend the 60 classes. Now decide like how many hours, how many days you will take to attend the 60 classes. It will be just, if you just watch 3 classes per day, just calculate like how many days you will be taking to complete the 60. So this is how much simple it is to cover genetics, because a lot of students always tell that genetics is not that easy, it is very complicated and it is not easy to understand. But if you start attending the classes and just taking the notes, so referring to the notes, everything will be simple. The students from the previous year, they already knew it. So next, apart from the Pathfinder, there is one more, like there are 2 more important books. So from the foreign author, which are like very bulky. So I would definitely not recommend you to read it if you are a beginner. This is Sanaster and Siemens, and another one is Tamarind book. So okay, this is the foreign author books. So there is no need to purchase these books because these are very costly. Like it costs around like 8,000 to 9,000, sometimes higher than that in the Amazon. But what you can do is like you can just search it in the Google. You will get the ready-made PDF of this. So ready PDF will be available. So if you do not want to read the complete book, if you want to only read the specific topics, if you are really interested, very, very interested in genetics, that you have extra time and also if, you have extra interest, then you can go for these 2 books. Okay. So apart from this, sir, like if there is there any other books that you recommend? So the one book that I would recommend is the master book that I would definitely recommend is like Handbook of Aggregated Genetics. But the thing is like it is not yet published. It will be published by December end mostly. So this book is a compilation of all these books plus whatever that we learned from our MSc PhD classes and whatever the students of IRA learned from their MSc PhD classes, the content of that plus from all this book compiled, the combined one, you will be getting it in the Handbook of Aggregated Genetics, which will be mostly published by December end. Okay. So this is about the complete book list. So if you stick for the single book and keep it simplified, you can definitely cover most of the things. Nothing complex required. See, as a senior, what I would definitely suggest is, before complicating yourself by gathering lot of books, go one by one. Okay, just take one book, try to finish it, try to understand the concepts that is available in that book. Then if you feel like very confident and if you feel that okay, I need some extra knowledge, I need to learn extra, I'm getting everything from this single book, I've almost understood everything, then try for the other books that I am recommending here. If not, stick to the first book. But since like there will be also some category of students who have already learned all the basics, are like waiting to know more about this genetics, you can go for the other books. Okay. So next thing, content outline. So if you know, like if you have already accessed the BDC book, so we have like tried to replicate same thing from our content outline, because if you see the previous year papers, like there are a lot of direct questions that will directly come from the BDC. Even the paper setters till today, till this time, they are using the BDC as a standard book to frame the questions. So that's why like we have prepared the outline and for every year, and this is our 4th year of plant sciences. So for this also like we have outlined the content from the BDC itself, so that whatever you read, the similar structure you'll be able to find it in our online classes. Okay. So first one is like genetics of history of genetics and cytogenetics, the first thing that we are going to cover today. And next, like theories of evolution. So in this, along with this theories of evolution, I will also try to cover the evolutionary genetics, which is actually not available in the BDC. This is something which is like connected with the theories of evolution. So if you like are able to understand it one go, evolution genetics and the theories of evolution, then your concepts will be fully clear. So I'll include it here. The third part is like cell ultrastructure, and then after that we'll go for the Mendelism. Like all the things that are related to the Mendel's work we'll be discussing in the Mendelism. And next, the application of Mendel's laws and testing of genetic hypothesis. So here you might find that this same terms are not available in the BDC. So this is from the some other book. So I felt like this flow is good, so that's why I use the term Mendelism and applications of the Mendel laws, testing of genetic hypothesis. Next, the gene symbols, the extension of Mendelism, and the chromosome structure and function, and the cell division, regulation of cell cycle, and chromosomal aberration structural, and the chromosomal aberrations numerical. So if you ask me, sir, why you have mentioned some of the topics like 2 times, okay, if you ask me like why you have mentioned 2 times, that's because this is exactly how I am going to take the classes. For example, for the numerical aberrations, for the chromosomal aberration numerical, I'll be taking the 2 classes. So that's why I mentioned it twice. Next, there is physical basis of heredity and the environmental effects and the recombination in fungi in the bacteria. So here, the 14.1 and 2, like I have not taken these classes in the previous year, but this year we will be going to clear it, cover it. So although I would not recommend you to go in detail from the BD Singh book, this chapter I always tell the students to skip it because it is not as such required for your ICAR PG exam. Plus, it's also complicated to understand. But I'll try myself to cover it in a very simple words. It's obviously because the year by year, as the competition is increasing, the students are well prepared. The paper setter will be also trying to give some difficult questions, so which are not included in the previous year. So considering that part, so we will be covering this in this year master course. Next, like inheritance of quantitative traits, linkage and crossing over in 3 classes, which is very, very important. And next, we'll go with the sex determination and again 3 classes. This is also another very important class for you. And next, the cytoplasmic inheritance and maternal effect, another important chapter, plus multiple alleles. So which is like most confusing, I guess. Like if you try to study from the book, few things you will be able to get it, but not everything. So that's why I tell it as confusing, but I'll try to simplify, need not to worry about it. Next, the mutation. Mutation is also another important part where you can definitely expect the questions. If the part is like, if the portion is genetics, you can expect the questions from the mutation. And next, like mutation detection. So in the plants, detection in the plants and gene editing, like we'll give you the basic intro regarding the gene editing. What is it? What tools are used in the gene editing? Because this is like now very burning concept, like where a lot of updates have been done in this gene editing, and every work that is related to genetics and plant breeding, it is based on this gene editing. And next, we'll also talk about the gene interaction, which is also called as epistasis. Then we'll jump into the most complex part of genetics, which is like molecular genetics. So molecular genetics will be covering genetic material, the double helix model, the DNA replication and repair in 2 classes. Next, the molecular techniques in genetics and the recombinant DNA technology. So same thing, these 2 were not covered in the previous year master course because of the same reason that they were not required, and the questions were usually not asked from this part. But considering the competition, so we'll try to include and make it simplified. So molecular techniques in genetics, which includes the markers and all, it will be dealt by Abhishek. So this is mostly of a plant breeding part. Okay, and this recombinant DNA technology, I will be covering it. Okay. Next, the organization of packaging and packaging of genetic material will be able to cover, and the gene action 1, genes, biochemical reaction and proteins. Next, the RNA processing, transcription, translation, protein synthesis, regulation part of both prokaryotes and eukaryotes, transposable elements, and the population genetics. So this is where we will be ending the genetics. Okay. So this is all about the complete structure of what we are going to cover from the genetics part, which will be mostly covered in the total class of 60. Someone is asking, yeah, one thing to clarify regarding the JRF, like regarding the ICAR PG and the PhD course. ICAR PG and the ICAR PhD course. So for this ICAR PG and the PhD, these classes are same. Okay, these classes are same, where all the basics will be covered in that. And after this, there are few things that needs extra for the PhD student, that will be covered in the advanced topic course that you can see in your course. So if you are enrolled to the ICAR PhD course, you will be getting one genetics and plant breeding course with the students of ICAR PG, and plus you will be also getting the advanced topic course. In the advanced topic course, we are going to cover what is not covered in this part, the basics. But sticking to the PG, all these things will be covered. So all the ICAR PhD aspirants also, if you want to cover the things from the basics, if you want that refreshment, you can attend my classes, or you can take the recordings, or if you are already very confident about the basics, your basics is strong, you are mainly focusing on the plant breeding, then you can directly jump into the advanced topic course. So mostly advanced topic course for the plant breeding will be starting by January. So till then, if you are already preparing for it, you can take the recordings that are already available from the previous batch. Okay. Yes, we will end all the questions related to the topic in the every class. Okay, we'll do it. So class timings, as I told you, like it will be like 10 p.m. mostly. Like if there is any change in the timing, I will inform in the master group at least 3 to 4 hours or 6 hours before the start of the class. Okay, fine. So this is about the class, and if you have, I have to like again simply explain about the class features. The first thing is live class recorded classes. All the live classes that we are going to take, it will be also available as a recording. Then we provide the complete study materials in the form of PDF that will be available within the mobile application. You can download within the mobile application and access it. And next, we provide the complete revision series after completion of the syllabus, which is something which is complementary for you people, where after the completion of the syllabus by April, we will be conducting 2 months of revision series and 2 months of rigorous mock test series to boost your preparation. And regular mock test, in this regular mock test, if you know already, if you have checked it, you have like topic wise. Now we are freshly introducing for your batch, the lucky batch, the monthly mock test, where every month end, whatever syllabus is completed, whatever classes are completed in that month, so that test will be provided in the monthly test. And after that, you have like full length mock test. See, the full length mock test will be only uploaded after completing the syllabus. Okay, before completing the syllabus, we will not be uploading the full length mock test. Only 2 mock tests, 2 full length mock tests will be available right now, so that you can understand the pattern, you can understand the level of difficulty. But all the other full length mock tests, approx there are 25 plus full length mock tests that are available with us. So that will be only available after the completion of the syllabus by March or April. Okay, along with this, the complementary is general agriculture. You will be also getting the complete general agriculture recorded classes in your course, in every course of the ICAR PG and the PhD. Plus, you will be also getting the mock test plus video lectures. Both the things you will be getting. It's upon you at which subject videos that you want to see, like starting from agronomy till the end. So you will be giving the video lectures for all the subjects based on your choice. Whether you want to watch it or not, you can do it. Plus, you can access all the mock tests, attend all the mock tests to test your knowledge. Next, short notes and crisp notes. You already know it that for the genetics, for the plant breeding, and for the seed science, there are some very, very important data that you need to remember it every day. On an everyday basis, you have to revise it. All those will be provided as a crisp and short notes, which is already uploaded in the course of the crisp note course, so that you can access from to day one. And if you ask me how to remember the memory-based content, the only mantra, the only solution that you have to follow to remember the short notes and the crisp, you have to remember that memory-based things is just revise it every oftenly, like on a regular intervals. Try to revise it so that in a long term you will be able to remember. That's the only simple secret. There is no other secret to remember these things. Okay. Next, previous year papers. So these discussions are already available till 2025. Sorry, 2024. So we'll be adding the discussion series of 2025. If needed, we'll be also again taking the previous year PYQ discussions. Next, doubt clearing sessions. If you know already that this is the one new thing that we have included from this year, which is doubt clearing sessions. So practically speaking, there is a practical difficulty for the students who like joined our WhatsApp group. Till last year, I mentioned that whatever doubts you get, directly post it in the WhatsApp master group. We will answer you. But there was a problem. The students were like posting the doubt, someone else was answering it. So we were also knowing whether the student is satisfied or not, and we were sometimes leaving it as it is. Sometimes we are replying on our end too. But students were hesitating that like they are not clarifying their doubt. But still, for this reason, what we have done is like we have introduced this doubt clearing sessions, where in every master group, every subject master group, there is a Google form. So first drop it in the WhatsApp master group. If you are not getting the instant answer, if you're not getting the clarification for that questions, if you need the detailed explanation for whatever doubt you are having, whether it might be the MCQ questions that you have seen somewhere, or it is related to any concept that you are reading from the textbook or reading from the notes, or any kind of questions, if it is you are not satisfied with the answer that you're getting in the WhatsApp master group, directly click on the form and fill the form and add the relevant images or the text. So every weekend we will conduct the doubt clearing sessions either on the Saturday or Sunday. So this will be available for every Saturday, Sunday, and mostly Saturday, Sunday we will not be having any class. There will be no regular classes, but we will be conducting this doubt clearing session so that on a video live class, your doubts will be cleared. So whoever has posted the doubt, they have to compulsory attend. Plus, all the other students can also attend the class, and this will be also available in the recorded form. And the PDF of what doubts you have asked and what we are answering, that will be also uploaded. Okay. So this is one additional new thing that we are going to do it from this class. Yes, for CUET PG also, like these classes will help, but I would say like for CUET PG, this will be like more than just help. Okay, it will be in depth. So I would suggest to all the students who are wanting to prepare for CUET PG, take one subject and prepare for ICAR PG exam, plus as a supplementary prepare for CUET PG, so that you will have two things on your line, okay, as a backup. Because for CUET PG, don't like it doesn't require like rigorous preparation. It doesn't require like in-depth preparation. Only most of the memory-based content, most of the subjects should be like read very regularly on a regular intervals. So that's what it requires. Only the general agriculture books that you all referring from so many years, that are sufficient for all the competitive exams. Whatever general agriculture books the students are reading, so that is sufficient to crack the CUET PG. Only it demands the rigorous revision, but you need not to waste like one year of your time, or 6 months, 7 months of your time just to prepare for this CUET PG exam, which do not give you the admission to the IRI or top other agriculture institutes, plus which do not have any fellowship. Okay, stick to one subject, take one subject if you are seriously willing to do the MSc, and along with that you can also prepare for CUET PG. That's not difficult. Add some questions. Okay, English classes. Yes, English classes will actually overlap with the Hindi classes. So if you are attending the Hindi classes, if not always it will overlap, but sometimes it can overlap. But if it is overlapping, you can take the recordings. For all the students, like if you are for the Hindi medium or English medium, you will be getting all the classes, or access to all the classes. Do not worry. If you wish to watch both the classes, you can. If you wish to watch only the single language classes, you can do it. It's completely upon you. Okay. Okay. Anything else? Notes. Regarding the notes, like last year students, till last year, what I did is like I gave my handwritten notes. Handwritten notes was uploaded till last year. But again, the lucky batch, your batch, like we have given the printed notes, which is already uploaded for genetics and plant breeding. For genetics, right? Printed notes will be available. Printed typed notes, sorry, not the printed one. So you will be getting the typed notes that are already uploaded in the course itself. Along with the typed notes, soon we will be coming up with the book, that is aggregated book. So first we starting with the genetics. Once we launch the genetics books in December, we'll start working on the other books. We'll try our best within March. All the subject books, all the other subject books will be also launched. Okay. So this is regarding the notes, the one update, and the mock test details. I already told you, we will get the topic wise monthly mock test, plus we'll also get the full length mock test plus general agriculture mock test all together. But the full length mock test will be only uploaded after the completion of the syllabus. Do not ask this repeatedly. Okay. So that's it for the things that I wanted to explain regarding the course. Now let's start the class. Okay. How's your energy? Energy is down, or it's okay? It's fine. So are you ready to start the class of genetics? Are you excited? Anyone? Both late ho gaya, sir. So you have told so many things, we lost the interest already. Is it so? Class, like we came to have a class, but it was only of introduction, not a class. है ना? Okay. So introduction सब कुछ. So let's stop the introduction and let's start this history part. Okay. थोड़ा blur लग रहा होगा. Okay, fine. It is fine. Okay. So first thing, the term genetics. So we always start with the history of genetics, and then where we start with the genetics term. So if you, like anyone, what is the term genetics? It is derived from which word? So what does it mean? What does the word genetics mean? Anyone comment in the chat box? Yes, quick. So it is derived from the term geneticos, which can be also called as genesis. And this genesis, if you break this term, and what does it mean? It is from, like, originating from. So this is what it means, right? So this is the term genetics, geneticos. Okay, fine. And also it can be also told as like to generate from. The two words, either to generate from or originating from. This is what the word geneticos, which can be told as genesis, the term meaning. And this is a very important term. It always starts with the term genetics. You have to remember this part without any mistakes in the spelling also. Okay. Next, after that, so the history. So how do we study the history of genetics? See, before going into the concept of genetics, it's obvious that we have to know about the history. What are the different events that have been taken in the field of genetics? Okay. So in this, for the ease of convenience of study, that how we can study this history. So it has been divided into 4 different parts. First one is like before 1860, and next it is from 1860 to 1900, and next it is 1900 to 1944, and lastly like 1944 to present. Sir, why it is being divided like this? So what is the reason for this? Before 1860, what it is called as? It is called as pre-Mendelian era. So my handwriting might not be very, very clear. So do not go with the handwriting. Just listen to me very carefully. Okay. So because if I try to write a very nice handwriting, the speed might be slow down. So next, like 1860 to 1900, it is the birth of genetics. So it is like Mendelian era, where the fundamental rule of genetics was established, right? So this is from the 1860 to 1900, and next the 1900 to 1944. So this is the post-Mendelian era. This is post-Mendelian era, and after this, like 1944 to present, like what has happened in this case, 1944 to present, the birth of the biochemical genetics, the birth of the molecular genetics, and whatever technology and the science that we have discovered till now, it comes all under this 1944 to present. But latest till now we will be studying till 2000. After 2000, there are no much like major events that have been taken, but still you can expect some of the questions related to the Nobel Prize, like the Nobel Prize recently was awarded to this and this scientist for what cause, like what for what discovery. So these things you have to list it out after 2000. Take it as an assignment. Till 2000, it will be available in the notes, but after 2000, what major things that have been done in the field of genetics? One thing I told you about the Nobel Prize, but apart from Nobel Prize, so many things are happening in terms of like recombinant DNA technology, in terms of study of genes, the study of like a lot of other genes have been disappeared. So like there are a lot of other things in that you have to pick up the highlighted points wherever if it is possible and keep it. That is extra thing, supplementary, that you can always go for. But other than that, so these are the basic things that every student who is preparing for this ICAR PG Plant Sciences or the PhD genetics should be knowing about. So that is what we are going to discuss. And in this events, I won't be like reading all the events. I'll be only reading, or I'll be only explaining the some of the important events. Rest you can study it by yourself. Anyhow, like if I start explaining all these things, like you will be getting bored. First thing, so the history you can start it like with the microscope. So how it started, you can start it with the microscope. So first microscope that was built was a compound microscope, and once the compound microscope was came into existence, then it was like modified, or you can say that it was upgraded by whom? By one of the very famous scientists into the simple microscope. And then after this, there are nucleus, the cell, the term coined, the cell was coined by the Robert Hooke in the 1665. And this simple microscope, if I have to say, it was like Antony von Leeuwenhoek. If you want to also know about like other achievements, like can you anyone just drop in the comments like what other achievements that or other contribution that Antony von Leeuwenhoek has made. So one thing to explain here, this spellings. So always remember that we as a student while reading, so we have that mindset that we neglect the spellings, but in the exams there might be the case where the spellings might be also evaluated. So ensure that whatever the important scientist names are, you should be able to write it as it is. Okay, and plus the complete name. For example, the properly working compound microscope, we know it, we already knew it, it is Jensen and Jensen's, but the full name. Just if you are not able to remember as such, but at least see it from your eyes, that will be locked in your eyes. So in the exam, if you see it in the options, exactly you can match. Okay. And this Jensen and Janssen, they were actually the father and son. I'll try to just cover the extra things that are not there in the book. Rest you can read it by yourself. Next, I told you like this was simplified by the Leeuwenhoek. And what are the other achievements that Leeuwenhoek made? Anyone? No one is commenting on this. What are the other achievements he has made? So I want you from your end. Next, after that, in the 1665, you know that term cell. It was coined by the Robert Brown by observing the cork. Next, in the 1974 to 1983, the discovery of the living organisms. So the Leeuwenhoek was the first person to observe the microscopic organisms like bacteria and the protozoa with the help of the microscope, the simple microscope or the modified one from the compound microscope that he has done. And what is this on the right corner? What is this image? Yes, animal term. Yes, good. This is the picture of the simple microscope that was prepared by Leeuwenhoek. Okay, so just see the term. This is very interesting. Next, it was in 1824 that Drucker concluded that all the biological tissues are composed of cells. So before you all know about the cell theory. Everyone are well aware of cell theory, but before the cell theory, there are other scientists also like who have postulated the same thing. And Drucker was one of them who concluded that all the biological tissues that we see, they are composed of the cells, and these cells are attached with each other. But the contribution of the Sleeden and Squin was very popular along with the Rudolf Uchu, that they come up with a concrete explanation for these things, so that they were awarded for the cell theory. Yes, this is very important. Very good, Vidya. That Leeuwenhoek. So this is how you have to study for the exams. Like if you see the term, all the related information regarding the scientist, you should be able to write it in the same. So who is he? He is called as father of microbiology. Okay. So since like he started his microscopic studies with the help of the simple microscope that he has built and observed all the bacteria and the protozoa for the first time. So he is known as the father of microbiology. Okay. Next, after Drucker. So one thing here, it is regarding nuclei. Okay. So in 1831, nuclei of the cell was discovered by Robert Brown in the cells of transgens kentia. But before that, Leeuwenhoek also done the same. So if you have read it, that Leeuwenhoek was the first person to observe the nucleus of RBC. So, but there is some problem with this statement. Can anyone tell me what is the problem with this statement? Yes, quick. So there is some problem with the 1674 where Leeuwenhoek was the first one to discover or observe the nucleus in the RBC. So it is about the common thing. It's the mammalian RBCs. Mammalian RBCs, mature mammalian RBCs lack what? They will lack the nucleus. So nucleus are not present in the mature RBCs of the mammals. But here, in which organism did he observe this nucleus? Yes, anyone drop it in the comment section. I won't tell you here. So that's the question. So that's always the confusion that so everyone reads a Leeuwenhoek observed the nucleus in the RBC, and later in some part in the book they will study that RBCs lack the nucleus, and then they come back and like confuse about like how did the Leeuwenhoek did it. Okay. Yes, lack of nucleus. Camel. Okay. Okay. Fine. So I wanted to just emphasize one thing here. So I told you that Jensen for the first time he was a person to discover or to build the compound microscope. And then I told the second statement that it was upgraded by the Leeuwenhoek and made it simple. So what changes he has made, and what were the features of the compound microscope, and what were the features of the simple microscope that was given by the Leeuwenhoek, which is help him. So has been given in concise way. So you can study this. Like for example, the magnification first one, it was very low, and then it was very high with the Leeuwenhoek simple microscope, and resolution was poor here, and it was superior compared to the compound microscope, and it was actually novel. But after it has been discovered, the Leeuwenhoek has made simplified by reducing the complexity and ease of use. Since it was very bulky, it was difficult to use, but this was tiny, and it was also difficulty because of tininess. So this is some of the key features and the key differences between the microscope that was built by the Jensen, and next the microscope that was given by Leeuwenhoek. But the Leeuwenhoek's simple microscope has actually helped in making the discovery, or making the discovery of the bacteria and the protozoa. Next. So after this, the cell theory. So who are the scientists who were credited for this cell theory, and why? So this is a very, very important questions. So when it comes to the cell theory, there are two things: animal cell and the plant cell. So we have already studied this from the class 11th and 12th. See, when it comes to the animal cell, it is Squan. Okay. When it comes to the plant cells, it is Scledan. So this is where like most of the students will be confused about Scledan gave the plant cell theory, or the Squan gave the plant cell theory, and how to remember it. So I apply the simple trick here. Squan, there is a, it is for animals. That's it. In this, there is no A, so that gives for the plants. That's how you can remember. So mostly like wherever there is a confusing terms, personally I use this trick. I find whether there are some matching letters in both. If there are some matching letters, I'll just match them. Like here in this case, it is for animal, and there is no matching letters, that will be for the plant. That's it. And the years are very important in 1838 and 1839. So one thing to remember, the cell theory was independently discovered, or independently given by the three different scientists. So this independent term is important. Anyhow, like seeing the term like 1838 and 1839, you understand, right? The logically you understand, but also remember that independently. This cell theory was given independently, and it has been credited to the three different scientists. So what was the contribution of the third scientist, the Rudolf Virchow? So in the cell lineage theory, when I talk about a theory called as cell lineage theory, can you tell me like what are the names of the scientists that you always hear? Do you always hear only Rudolf Virchow, or is there any other scientist that you also see in the books? Yes, quick. Any other scientist? Anyone? Board is, so the board is not clear for everyone? Yes, exactly. Very good. It's Nageli. Just confirm me once, like if the PDF is clear to everyone or not. Okay. Vidya, just check your internet connections. It might be because of the connection. So one thing here, it is regarding the Nageli. So there will be always a confusion whether it is a Nageli or the Rudolf Virchow. See, in order to clarify this, Nageli just given the postulation regarding the cell lineage theory, but the detailed histological studies were done by the Rudolf Virchow, which gave the concrete evidence for this theory that yes, the cell lineage theory exists. So according to the cell lineage theory, what if I have to expand it? What exactly this means? Cell lineage theory. Lineage. Lineage is something that has come from the ancestor to descendant. How something has originated. Talking about the origin. So talking about here, they are talking about the origin of cell. How the cell have come into existence. It is from the pre-existing cells. So all cells will come from the pre-existing cell. That is what given in the cell lineage theory. Okay. So there are three important postulates of the cell theory according to these three scientists. So first thing is like all living organisms is made up of cell, and the cells are what? They are structural and the functional unit of organism. And third, all cells come from pre-existing cell. Okay. So this was also called as cell lineage theory. That was given by the Rudolf. And first two, they are given by the Scledan and Squan. Here, while remembering the term, while remembering the scientist, the Skledan and Swan, it is also important. Just try to remember the full name. Don't know like if it will come in this exam or not, in this year exam or not. You can definitely expect. So at least know the Matthews, Jacob Skledan and Theodore Swan. Remember them as a full name. At least some of the very, very important and very, very common scientists, at least you should be knowing for a full name. Next thing, the cell theory, I told you, so 1839 and this last thing, 1855. Okay, so this 1838, 39 and 55. So this you have to remember. The Rudolf Virchow cell lineage theory in 1855 and in 1838 and 39 about the cell theory. Exception. So next thing, there is one very important exception to the cell theory. What was explained in the cell theory? So what is the exception? Yes, quick. You have to mention it quickly. What is the one exception to the cell theory? Quick. Arey, why are you taking so much of time? What is the exception to cell theory? Does all living organisms have cells? Do you agree with me? Is this statement correct? Yes, viruses. That's it. Like I was expecting a simple answer that it is except virus. Why? Why it is there is exception for the virus? Because virus, although they are not living, they are, and also they do not have any cellular organization. There will be no cellular organization in the virus. If you just see the composition of the virus, what it will be? It will be just the nucleic acid plus what? Protein. So the nucleic acid can be what? Either it can be the DNA or the RNA as a genetic material. So this is a simple composition of the virus, which although it is like between the living and non-living, it is an exception to the cell theory, that it is not under this thing. Okay, so this is what you have to remember. Cell lineage theory, in this there is a one very important sentence in that is given in the Latin word from the Rudolf Virchow. What is it? Can anyone type? So the Latin sentence, it's not a word, it's a sentence. Let's make it a sentence. So which was translated into the all existing pre-existing cells are made up of cells. Sorry, all cells originate from the pre-existing cells. Yes, omnis cellula e cellula. Yes, very good. So the term, the Latin sentence, you can remember this like omnis cellula e cellula. Okay. So this is translated to what? All cells originate from pre-existing cells. Okay. So I'll tell you again. Please neglect my handwriting. Just listen to me very carefully. Yes, live classes आज से ही start हो चुकी है. Next. So this is about the 38, 39 I mentioned, and in the 1859, the publication of Origin of Species. Next event, it was done by the Charles Darwin. So going for the series of events, like we came till the 1838, 39. After that, in the 1859, one more important event that came into existence is like Origin of Species. The publication of this series with the also by giving the one of the very, very important and the famous theory, which is like theory of natural selection. It was given by the Charles Darwin. Okay, it was in the 1859. Next, we have just completed the most important events from the pre-Mendelian era, where we started with the microscope and then we got to know about the cells, and the cell theory was the major highlight in the pre-Mendelian era. So after that, what about in the Mendelian era? You know it already that most of you, if you are coming for the plant sciences, you at least know this, that in the 1866 the publication of the Mendel work was done. So in which paper it was like in which publication it was published, and what was the title of the paper, and what was the details of the paper, that everything will be discussing in the Mendelism class. For now, let's stick to this event details. We will not go into details. And in the 1871, there was the first time the technique for isolation of nuclei was done by the mixture, and it was called as nuclein. So how the nuclei should be isolated, it was given by the mixture. He did it for the first time and he called it as nuclein. But later it was changed, I'll tell you. And next in the 1875, it was Hartwig given the fusion of, explaining the fusion of sperm and eggs. Later in 1878, describing the chromosomes. Who described the chromosome for the first time? It was for Walter Fleming. And who gave the term chromosomes? So there is a lot of confusion between some of the scientists about the chromosomes. So can you just list out or type it in the chat box? What are the different scientist names that you usually read about when you talk about the chromosome discovery? What are the different scientist names that you get in your mind or you get always confused with? So Walter is the right answer for the term chromosomes. Very good. But if I have to ask you about like other names of the scientists, you get for with along with this. Yes, it is like Strasburger. So students will always get confused with the contribution of the Strasburger, contribution of the Walter, and the contribution of the Walter Fleming. And there is also one more scientist. Who is he? There is one more scientist here in this line. Is like Nageli. So I'll describe all the contribution of this scientist in a single slide. Next, in 1882, the Walter Fleming proposed the term the mitosis. See, whenever you see the mitosis, you can definitely go forward to the meiosis and also know this. So meiosis was given by whom? There are two scientists who gave the meiosis. It was by Farmer and Moret. So this is how I would suggest you to read. It's not like you start from one end and go to the next end. You, while reading it, so if you get any relevant term which is related to the something that you are reading, you have to immediately recall the other term and the following contribution who made it. So that's how it will be simple for you to remember the things. And in the 1889, the nuclein was converted into nucleic acid. The term was converted and it was by the Altman, which is now universally accepted term. Okay, it is now called as nucleic acid, and this was given by the Altman. And next in the 1890, the X chromosomes were discovered. It was by Enking. You know this term, this scientist like the Enking. He was the first one to discover the X chromosomes. Okay, and he was a German biologist. Decoding the mystery of the chromosomal discovery. So there are 4 different years. Similarly, like there will be 4 different scientists, and the 4 different scientists I have given the 4 different things related to the chromosomes. So I will just list out the scientist names. Try if you can say something about this. First one is like 1842, it was Nageli, and in 1875 it was Strasburger. Next, it is Walter Fleming. And lastly, you know it already, it is 1888, it is about Walter. So, can you just tell me like with the Nageli, so this is also very important. He gave or he explained the chromosome to be, he observed something in the cell and he explained that as a transitory cytoblast. So remember this term. This was given by whom? It was by C. von Nageli. Okay. Yes, very good. Someone has mentioned it. What is that? I'm not able to pronounce your name. Okay, let it be. Okay, transitory cytoblast. Very good. So next, Strasburger. Strasburger was the first person to describe the chromosomes to be the thread-like structures. And so then what is the contribution of the Walter Fleming? So Walter Fleming clearly described the chromatin later. So it is just like the, what do you call, the decondensed form of the chromosomes, the chromatin, the loosely packed one. So that he observed and he described this chromatin majorly. And lastly, the Walter, what he did, it gave the term chromosomes. So this is how you can simplify the confusion whenever you see the 4 different scientists for the single term, which is the chromosomes. Just remember this point. So this has been clearly given in the notes that is uploaded in the course. So you can write it down somewhere, and regularly if you see it, so you'll be able to clearly describe and differentiate between the 4 different scientists. Although I have just only briefed about their contribution, you can also read it in detail about them. Next, the post-Mendelian era from the 1900 to 1944, and the rediscovery is the one of the major event from the post-Mendelian era. So if I ask you, like, obviously, like, everyone will tell it, I guess. So the three different scientists who were responsible for the rediscovery and who were credited for the rediscovery. So can you just name them quickly? It's very important that you know the complete name and where they are from. Okay, you can't miss it. Hugo de Veres, Carl Correns, and Erich von Schermach. So these are the three important scientists: Poland, Germany, and Austria. So all these are important. So you have to remember from the 1900. So this was just the rediscovery of what Mendel has already published in 1866. So once we study the Mendelism, so we will in depth deal in these things. Okay, fine. Okay, some people are answering. Very good. Okay. So next, in the 1903, so next important event after the rediscovery was the chromosomal theory of inheritance, which most of you might have already known about. The chromosomal theory of inheritance. So even with the chromosomal theory of inheritance, there are like three scientists' name that you usually see and you usually confuse with. So who are the three different scientists for this chromosomal theory of inheritance? Yes, quick. So two are already present here, like in this slide only. The Boveri is here and the Walter Sutton is here. Along with this Sutton and Boveri, who else you usually read for this chromosomal theory of inheritance? Yes, Sutton and Boveri is correct. Walter Sutton and Boveri. Okay, what the other name? The other name. Do you not study like the Thomas and Morgan or T.H. Morgan whenever you study about the chromosomal theory of inheritance? Yes, it is 1902 and 1903. This is also like independent work. So done by the two different scientists in 1902 and 1903, Boveri and the Walter Sutton. So this here, first the theory was given based on the explanations, the different experiments and explanations. The theory was regarding the chromosomal theory of inheritance was given by Sutton and Boveri. But the experimental evidence regarding this, that genes are located in the chromosomes, that was given by Thomas and Morgan. So before that, if I have to ask what is the connection between genes and the chromosomes, what do you answer? How do you relate these things? How do you explain these two different terms? What is gene and what is chromosome and how they are linked with each other? So this linkage is like the complete explanation regarding like how genes are linked with the chromosomes, how they behave together. This was clearly defined in the chromosomal theory of inheritance, which explained us, which got us to know that genes and the chromosome behave similarly. Okay, so that's how it was discovered that genes are located in the chromosome itself. So chromosomal theory of heredity. So same thing, the inheritance and heredity, don't get confused. It can be heredity or the inheritance. So they are not different. The physical basis of heredity was explained by this chromosomal theory only. Okay, and anything to add here? So 1902 and 1903, like if I have to tell you about like what Sutton and what Boveri did. So in 1902, so Boveri gave the experimental evidence. So it was like experimental evidence for what? So he gave the experimental evidence that specific chromosome will have a specific role. While studying the C. urachin for the fertilization event, you try to fertilize with the different cells. So where he found that where there are different cells with a different chromosomal count, so there was a different effect that was produced in the after the fertilization in the embryo. Somewhere like fertile, somewhere not fertile. Somewhere normal, somewhere abnormal. So finding this, he discovered that so there are specific chromosomes which have a specific role. So that was discovered by the Boveri here. Okay, and then what does the Sutton did in the 1903? So he did the cytological study. What is cytological study? It is like the studying the cell division, the proper cell division studies he had done, and he has observed that the characteristic of the genes, like for example, genes has a two alternative forms like alleles, right? It has in the one pair, the two, and the chromosomes also come with pair, and they divide during the meiosis. The similarities between the genes and the chromosomes was well studied by the Sutton in 1903. So these studied, compiled, like this compiled combination of these studies gave us the understanding that yes, the genes are located on the chromosomes, and that is why they're credited for the chromosomal theory of inheritance. Okay, fine. Next, so the term meiosis, the meiosis term was given in 1905. And what about the mitosis term? Can anyone tell me, like if you remember from the previous slide? So who gave this? Walter Fleming. So you will tell the scientist name. What about the year? So when was the term mitosis was given? Yes, I told the meiosis was given in 1905, but previously I told about the mitosis also. So when it was? It was in the year. Yes, anyone? I'm expecting answer from you. Quick. Yes, it was in 1882. So this is how you have to correlate. That's it. Okay. This is how the things get simple. Wherever you see the related terms, just go back to the previous one, recall it, rewind it, so that you'll be able to remember. So next, it is like 1905. Nettie Stevens, like American dentist, named the X chromosomes during the research in the mealworms. This is like larval stage of a beetle, which is called as darkling beetle in the Coleoptera. It was in 1905. Next, so it was also along with this, the Stevens was a person. She's a like she's female. Okay, don't get confused. She was a person who described the role of X chromosomes in the sex determination. So while we study that sex determination in detail, we'll understand in detail about like what are the different forms of sex determination, what are the different chromosomes, like how they are responsible, and how actually the sex is determined in the living organisms. That will be dealing in detail in the sex determination chapter. Next, 1903, the very famous theory, which is like pure line theory, was given by Johnson. And this, I guess, like it is a very common question which is asked in any agricultural exams. You should be knowing like Johnson, and you should be knowing like he has also given the term gene, genotype, phenotype, and he proposed the very famous theory called as pure line theory, and it was in the princess variety of Rajma, which is like Physalis vulgaris. So this considered the most common questions that will come in any exam. So do not like forget it. That's it. Next, the 1906, it was the first time. Again, when I talk about the multiple factors, so there are two scientists that you always get. One is Yule and other one is Nielsen Ellen. So in this, the idea initially was proposed by this about the multiple factors by Yule, but in detail, it was demonstrated by Nielsen Ellen, and it was through the studies of seed color in the wheat and oats. So what are these multiple factors, and how they have done it in these two different crops, the wheat and oats, that we'll discover once we come up this in detail. Okay. Next, 1910, the one of the very major important event. For the first time, the gene was discovered to be precisely present on some chromosome, was done by the T.H. Morgan. So which was the very first gene that was discovered to be present on a specific chromosome? Yes. Who described meiosis? Generally, the meiosis and the mitosis, the process, the cytological studies, it is not credited to a single scientist. Like it will be the series of events, series of research that the scientists were able to decode, like how exactly the cell division takes place. Okay. Yes, it is like white eye gene. So this is it's already present here. The white eye gene of Drosophila, the first gene to be located on a specific chromosomes. In which chromosome it is located? So this is for the Drosophila, the white eye gene. It is located in which chromosome? It is in the X chromosomes. Always student get confused. Like if a simple question comes in the exam that the Y gene is located, W gene is located in, again students will again get confused. So do not get confused. It is in the X chromosome. And the T.H. Morgan, what are the other achievements or the contribution? The first evidence for the chromosomal theory of inheritance. I told you the first evidence for this theory. Do not get confused. Yes, although it was postulated from the several different studies from the Sutton and Boveri, but the first conclusive evidence which is accepted universally was given by the T.H. Morgan. And this whole study gave the phenomena of the sex linkage. Okay, so all the study that he has done in the W gene, that is white eye gene of Drosophila, gave us a phenomena called as sex linkage. And what is it? So again, we'll explain in detail in the later classes. Next, in 1913, the first genetic map of the fruit file, the same Drosophila which the T.H. Morgan was studying, the same species, so was done by the Alfred Stuart Went. And who is this Stuart Went? Stuart Went was a student of T.H. Morgan. Okay, in his lab only, he was able to come up with the first genetic map. So when I talk about the genetic map, what is this genetic map? It is like a linkage map based on the recombination. So he prepared the first map of the fruit fly, which is Drosophila. And what is this genetic map? What is this linkage map? We will discuss everything in detail in the later classes. Don't worry about this right now. And next, in the 1927, the mutagenic action of X-rays. So this is again one of the very common questions that comes in any of the exam. For the first time, that mutation, the term mutation, if you know, it is just altering the genetic material, which will have some effect. This process can be induced by some of the agents. For the first time was discovered by the H.J. Muller with the physical mutagenic agents like X-rays. For the X-rays, he gave it in the Drosophila. He observed and studied in Drosophila that like subjecting the Drosophilas to the X-rays gave the mutagenic effects. So that discovered the mutagenic effects of the X-rays. But in the plants, for the plant breeding, which was very important, Stadler did it in the maize and the barley. But here, one important thing to remember, what? It's not only with the X-rays that he has worked with. What is the other physical mutagens that he has worked with, or the other radiations? Yes, along with this X-rays, what are the, which was the other one which recently was asked in the exam, which confused a lot of students? Yes, it is the gamma rays. So it is not generally mentioned in the book, but you have to specifically highlight this point. So it was in the both X-rays and the gamma rays that he studied for both wheat and barley. Okay, very good. So next thing, the Garrod, the inborn errors of metabolism, the first event, the first, you can say that not the first event, but first very important event that gave the birth to the another branch of genetics, which is like biochemical genetics, was this inborn errors of metabolism. So this was given by Garrod and concluded that, what he concluded from this study, like he concluded that a person suffering from hereditary disease, it might be because of lacking of specific enzymes, which might have not produced the protein. Because of this lacking, the person is suffering from a disease. That it is like how the gene is linked with the enzyme. So if the gene is not working, it will not produce the enzyme. If the enzyme is not produced, the protein is not ready. If it is not ready, it will have the effect on a phenotype. So that's how it was slowly broken down and understood. And in the first time, like 1941, the evidence to the above hypothesis. The hypothesis was, what was the hypothesis based on the Garrod? So based on the Garrod publication, hypothesis was one gene, one enzyme hypothesis. That every gene produces an enzyme which will have a phenotypic effect. So this hypothesis, the evidence for this was given by the Beadle and Taunton by making the studies on the common bread mold. What is it? It is Neurospora crassa. And they propose that each gene produces the specific enzyme. Okay, so now I will ask you this question: one gene, one enzyme, is this universal? This is true for all the genes or not? Yes, okay, it is also, you can, from the Garrod, you can interpret it as like one gene, one metabolic block. Okay, since there was a metabolic block in the complete pathway of the metabolism, there was a block because of a lack of specific enzyme. So you can mention it as one enzyme, one metabolic block based on the Garrod work. Next, what evidence the Beadle and Taunton gave? They gave this hypothesis of one gene, one enzyme. Okay. Now tell me, so it is universal. If you say no, then what is universal then? What is the universal term related to this concept? The biochemical pathway concepts related to the gene. Quick. Anyone? Polypeptide. Okay, good. So second thing was like one gene, one polypeptide. Again, I'll ask the same question. Is it universally true that all genes produce like all the genes produce a polypeptide? Polypeptide is nothing but the protein, right? One gene, one polypeptide. Is it true that it is universal that all the genes in all the organisms produce the same? No. Then what is the universal thing that is currently accepted? That's what I'm asking you. We started with the one gene, one metabolic block. Next, one gene, one enzyme. Next, one gene, one polypeptide. So what is the why can't we accept that this is one enzyme, one polypeptide? Why? Because the gene can also produce the RNAs. Yes or no? RNAs are proteins. RNAs are protein. And tell me one thing, all enzymes are protein? All enzymes are protein. There is a one very confusing terms. All proteins are enzymes. Which one is true in this 1 and 2? All enzymes are protein and all proteins are enzymes. Which one is true? Many polypeptides. Okay. If how it is one or many, fine. But don't you think like the gene also produces the RNAs which are not acting as the enzymes or which are not converting into the proteins? Ribosome exception is there. Yes, very good. The ribosome is an enzyme which is like all enzymes are protein, true. Okay. Okay, all enzymes are protein, true. How come? How it is? First one is true. Ribosome is the exception for what? Just think before answering. Okay, this is a homework for you. So whoever is watching this video, you can comment on this chat box. I need like correct answer. Take some time and answer this correctly. And here like one gene, one polypeptide. Why it is not true? Because not all genes provide the polypeptide or not all genes produce the protein. Some genes also produce the RNAs. RNAs also have the crucial function in the cell, right? In the metabolism. So that's why like the most accepted one is like one gene, one what? Tell me. I have to write it nicely, right? One gene, one diffusible product. This is the most accepted one. Diffusible product can be the RNA or it can be the protein or it can be the enzymes. All plant proteins cannot be enzymes. Okay, good. So this one I need answer. I'm just trying to confuse, but don't get confused. But still, I want answer for everyone. Just comment on this video, okay? After this video, like live ends. So next, so we were here, and next after that, I told you like this one gene, one enzyme hypothesis. This was the foundation to the start of a new branch of genetics, which is like biochemical genetics. And next in 1946, the Liederberg and Tontum. What was the contribution of the Liederberg and Tontum? Yes, I want you to answer this question. In 1946, there was a one important event related to the bacterial reproduction or bacterial genetic material exchange or bacterial recombination. It's like bacterial not reproduction, it is bacterial recombination. Yes, yes, exactly. Bacterial conjugation. So they were the first one to describe and discover this bacterial conjugation process. Okay, so this is like Joshua Liederberg and the Edward Tontum. Okay, next the Zinder and Liederberg, the same Liederberg with the Zinder. So Liederberg was whom? Like Liederberg was actually the student of Tontum only. So after that, they two collaborated and came up with the bacterial conjugation. And next, the Liederberg collaborated with the Zinder. They came up with the another important discovery. What is it? It was in the 1952. Yes, exactly, it is transduction. Very good, very good. So the same thing you will be studying in the microbiology too. So this is like a brush up and refresh for you, that you will be knowing the things from the genetics also and from the microbiology also. So here, so Joshua Liederberg, Tontum, and the Northern Zinder and Liederberg. So this was a contribution of these two. Now, if I ask you, the Tontum here, he and he are same or different? Same or different? They are same or different? Am I confusing you people? Don't get confused, just think. I don't know like whether you have given a thought of it, thought about it not, because everyone know the term, like everyone know the scientists like Beadle and Tontum, just Liederberg and Tontum and Zinder and Liederberg, but they are same or different? So they are same. Don't get confused, they are same. First it was with the collaboration with Beadle and Tontum, they gave up with the very important hypothesis, which is gave the understanding, the initial understanding about the metabolic pathway. So what I mean by the metabolic pathway is something like A is produced, how the B is produced, from B how the C is produced. So this is a pathway. For this, like there will be one enzyme here, the first enzyme. So this will catalyze this conversion of this one to this A to B, and if there is a second enzyme which is actually present, and this will catalyze the conversion of B to C, and there might be like other one which can catalyze the C to D, and D might be the final product. So this is a metabolic pathway. So here they describe that every enzyme here, it is coded by a gene, specific gene, and because of this, if this gene is not produced, or if this gene is not producing the enzyme, this metabolic pathway will be blocked. So that is what like one gene, one metabolic block means. Okay, and this was first given by Bitter and Tontum. After that, they came up with the bacterial conjugation, the Liederberg and Tontum, and next Zinder and Liederberg came up with the other one, the recombination in bacteria, the other process transaction. So along with this two, there is one more type of recombination in bacteria. Which is it? Yes, quick. There are three important. Yes, quick. At least answer this question. This is very easy question that I'm asking you. Yes, it is transformation. Just remember the transformation. So who gave this transformation? Just like I told you, you have to relate the concepts. If you are studying the conjugation, if you are studying the transduction, immediately you have to recall the transformation process and also its discovery. So who gave it? Yes, it was from the Griffith experiment. Yes, it was by the Griffith who gave this transformation. So coming back, now I told you first thing, I made it clear that they both are same. But one complex thing has happened here. The Liederberg, Tontum, and the George Beetle, all three got the Nobel Prize for this discovery. The key discovery was one, it was related to the one gene, one enzyme hypothesis, which led to the understanding of biochemical genetics, the initial novel discovery, and next it was the bacterial conjugation and translation. But one thing you will definitely ask me, sir, that Tontum was awarded, Liederberg was awarded, and the Beadle was also awarded. But why not the Zinder was awarded for this same? Because he was also a part of this, the transduction discovery, but why he was not awarded? Any answers? Just like in the Willikens, you always know this, that in the Maurice Willikens, Rosalind Franklin, and James Watson, and Francis Crick, out of 4 scientists, only the Nobel Prize was awarded to 3, and in every time in most of the, like in the previous year exam also, it was asked in the exam. So why the Franklin was not awarded with the Nobel Prize? Similar question I'm asking here, which is not very famous. It will become famous once you get it in the exam, and let's see, like in the next year exam it might come. Not actually, like don't. I should appreciate that at least you recall the Rosalind Franklin, that she was not alive, but same thing doesn't apply here. There is some difference. So first thing, the golden rule of the Nobel Prize, the rule of 3. You know the rule of 3. If you do not know it, remember the Nobel Prize will be maximum awarded to only 3 personalities, like 3 different people. Female, male, anything is fine. Any combination is fine, but only 3. It is a rule of 3 for the Nobel Prize. Okay. So because of this, what you can get this question, then what is the difference between the Liederberg and the Zinder? Why it was given to the Liederberg, not for the Zinder? First reason was like because of rule of 3. that it has to be given for only 3. So whom 3 to consider? So leader was considered because he was a part in both the bacterial conjugation and also with the transaction, having the heavy weightage compared to the Liederberg and the Zinder, and might be because of some other reasons. Like if you see the literatures, there might be like different speculation regarding this, but I consider this is one of the major reasons that Zinder was not given the Nobel Prize, and it was only awarded to the 3 scientists for their novel work in the field of biochemical genetics. Okay, so this is what you can remember here. I always try to give something extra that is not there in the book so that it keeps you interesting, motivated to study this branch of genetics. So take it as an informative. And in the exams, I don't know like whether you can get it or not. If you get it, try to answer it. That's it. Next, the last part, from the 1944 to present. So 1944, the very, very important event that has done was this, that started with respect to the genetic material. In the Mendelian era, people were worried about like how the traits are passing from one generation to another, how I am black, how someone is white, how is dark, how is short, how is tall. These things were in the complete discussion. But after that, the next thing that came into existence after the Mendelian era was the post-Mendelian era, knowing about the pathways, like how the gene was responsible for giving the traits. And after that, after knowing all these things, the next biggest question was like, what is this gene? What does it consist of? Like, what is it is made up of? And how the gene will give the enzymes and how it comes into the protein. What are the different processes? That's what the molecular genetics is. So in order to study that, the very first question was, okay, we are trying to talk about the heredity material, but what exactly is heredity material? What is the composition of this heredity material? And in the cell, there are different types of things, the organelles that are available, but which exactly is the heredity material? So that was the biggest question. So that started answering with the discovery or with the evidence from the 1944, the first time. It was started from 1928, I can say that, because it was from the Griffith experiment only. The transferring principle came into light, and after that, scientists came in the race to identify the genetic material, which is the genetic material. So along with this, including the second important discovery was 1944, where the genetic DNA was shown to be genetic material from the ivory, McLeod and McCarthy with the same experimental material what Griffith used, but the difference was they did it in an in vitro condition. So Griffith tried it with the mice, right? But these scientists, they did it in vitro, tried to do it in vitro, replicate it, and they concluded that DNA is genetic material. But because of few lacunas in the experiment, it was not clear that DNA is genetic material from the experiment of Avery, McLeod and McCarthy. So I'll tell you few reasons once we jump into the molecular genetics. For now, leave it. But the concrete, the universally accepted evidence was given in 1952. Who gave it? That DNA is the genetic material in most all the living organisms, except like you just skip the virus. But apart from virus, like DNA is a genetic material. Who gave it? Array, don't tell Watson, please. Alfred, full name. I expect always a full name. Whenever you at least tell the very, very important scientist name, you have to come up with the full name. Someone has Monica. Yes, Monica, just I want the full name from you. Very good. You answered it right. The full name, Alfred Hershey and Martha Chase. Okay. So they gave the conclusive evidence regarding the DNA is the genetic material, which led to this new branch. I can say like after that, once the structure of DNA was done, that the DNA was described, that was in every literature, there is a point that when this particular event has been done, that marked to the birth of the molecular genetics. That will come in the next part. So in the 1952, in 1950, one more in between this, like there was one more important event. If you know, the transposable elements, or also called as mobile elements. So they were discovered in the maize, which were also called as jumping genes, which are ACs and DS, the associators and dissociators. See, one very important and interesting thing that you all should be knowing is, see, whatever important discoveries that we have known or which we have read in the genetics, mostly they are from the prokaryotes, the simplest organisms, where they are like very ideal experimental materials to discover something. But the one exception, like one major exception, is like the transposable elements, which have that lot of importance. But it was for the first time discovered in the eukaryotes by the McClintock. Okay, so this is one interesting thing that you can remember. So next, in the 1953, so the one another very important event which led to the birth of molecular genetics, the DNA double helix model, how exactly the structure of DNA is, which was decoded by James Watson and the Francis Crick. Okay, along with this Crick and James Watson, who else is credited for this work? I already told you it was Wilkins. And why the Rosalind Franklin was not awarded, even though she had a significant contribution in this work? What is her contribution? What is their contribution? We'll talk in the molecular genetics. But for now, why she was not included in the Nobel Prize? Because she was dead. That's it. She was not alive when this was announced. So that's why they were not considered. Again, if she was alive, there would be like a lot of controversy again to whom to give this Nobel Prize, because there is always a rule of three they have to maintain. So this event never occurred, so we'll not talk about it. But even you can give a thought. If you read all the literatures, if you know properly that what was the contribution of 4 different scientists, you can come up with your own answer, like who should have received the Nobel Prize. In 1958, what was the another important event that was done? Another important event with respect to the genetic material. 1958. Yes, quick. Yes, Matthew. Very good. So, Suchitra, like you come up with the full name. Matthew Mathelson and Franklin Stahl. What was their contribution? What was the contribution of Mathelson and Stahl? DNA replication. So that is semi-conservative. Yes, they have given the explanation regarding like how the DNA replication takes place. There were 3 important models, you know it. It was conservative, it was dispersive, or it is semi-conservative. People were having the questions, the scientists were having the questions that whether it is like dispersive, whether it is conservative or semi-conservative, and it was finally like with the evidence provided by the DNA for the DNA replication by these 2 scientists, that DNA replication is semi-conservative. Okay, so this is another very important event in case of molecular genetics part. Next, 1962, along with the Wilkins, the Nobel Prize, it was in 1962 awarded, which led to the formation of this same genetics, molecular genetics. And 56, from here you can consider this is like all about the biotechnology or recombinant DNA technology. Next, after understanding the genetic material, the scientists started manipulating the genetic material. So this is where it starts, like recombinant DNA technology. It is from the 1956. So OCHA, first in vitro synthesis of RNA, sorry. First in vitro synthesis, like RNA was synthesized in the lab for the first time in 1956. And the Kornberg in vitro synthesis of DNA was done in the lab. So this was the first important event with respect to the recombinant DNA technology, the manipulation of the genetic material. Next, in the 1961, you know about the genetic code, the language between the gene and the protein, the genetic code. For the first time, Marshall Nierenberg, the main person to decode this phenylalanine, which is composed of uracil, which gives the code for the phenylalanine in a genetic code, the genetic language, the code language. So every 3 amino acids, like you know, if you know like genetic code is triplet, it consists of like 3 different bases. So here, this base gave the term the amino acid which is phenylalanine. The first code of this was given by this Marshall Nierenberg, and this actually gave the amino acids. Like this code language gave us the amino acids, then later the protein. So this is regarding the 1961. And in the it was in 1961. In the same year, the one important concept, it is regarding the operon concept. So if you know about the operon concept, 1961, the lac operon. It was by Jacob and Monod, that you have to remember, the Francis Jacob and Jacques Monod. So they gave the very, very important concept called as operon concepts, where they have explained how the structure of gene, what different components does the gene will have, and how it will give us the enzyme and the protein. So this was regarding the operon concepts. It was also a very, very important thing which we'll be discussing in very detail, since it is very important. Next, in 1970, Argobind Khorana synthesized the known gene. He, for the first time, synthesized a known gene which was coding for alanine transfer RNA, which is alanine tRNA. So gene which is coding for this RNA. I told you, gene will not only code for the protein, it will also code for the RNAs. RNAs, again, if you know, there are several different types of RNAs, in which, like, major types, if you know, the mRNA you have, like you have tRNA, you have rRNA. So these are the major types of RNA. So tRNA was one of the major. So how the tRNA is produced? Again, it might be because of some gene. So this gene which was coding for this alanine transfer RNA was for the first time given by or discovered by this Argobind Khorana. And in 1970, so Smith and Athens and Arber gave the first endonuclease isolation. Okay, so endonuclease isolation, what is this? Endonuclease enzyme, an enzyme which can cut the DNA. So that is what endonuclease. Endo, in the sense, if it is the DNA, there are two terms. One is exonuclease and other one is endonuclease. So what is the difference between the endonuclease and the exonuclease? Enzymes, the exonuclease and endonuclease. If I have to interpret in terms of like DNA structure, endo, it can cut the DNA anywhere in between. In between, it can cut anywhere the DNA in any part, but the exonuclease, it can only cut the ends of the DNA. At the free end, it needs the free end to cut it. Okay, so that's the difference between the exonuclease and endonuclease. And the first endonuclease, can anyone tell me like which was the first endonuclease that was discovered? Yes, the first endonuclease which was discovered. It is like type 2 restriction enzyme. If I have to give you the clue, it is in the 2. Actually, this is where the recombinant DNA technology starts, where the actual manipulation starts, not in the previous one. Okay, not actually at this part. This was just not here, just don't get confused. From here, the events occurred where the scientists started to manipulate the DNA. Before that, it was all about the understanding the DNA, the concepts of DNA, and plus understanding the mechanism of the gene action. Okay, just correct it. And next, in the 1997, the first mammal was cloned. The cloning was also started because of this recombinant DNA started, and the first mammal to be cloned was named as Dolly, the sheep. It was a sheep called as Dolly in the 1997. And in 2000, there was a human genome project that was completed, where most of the genes were decoded. The complete gene sequence was known. It is like an 2000. And there is one name, the one important name, person name, which is related to the human genome project. So can you name it? Name him. The very important name for related to the human genome project. Yes, it is Craig Venter. Very good. That's it. Okay. Yeah. So this completes the complete history of genetics and cytogenetics. I hope I try to keep it very simple. I try to explain the important events. I hope I tried not to bore you people by reading the things that are available in the slides. Although like we use the PPTs, we use the PDFs for explaining the things, but we mostly try that we do not just explain whatever there is a slide, because you yourself can read it. We try to give some value addition to whatever content you already know it. Try to gather some more information which you do not know it, and try to break the concepts which are very complex. If you have liked this session, if you have enjoyed this session, I want one heart from everyone in the chat box. Yes, quick. If you are not giving a heart, that means like I'm not doing well, and I will be continuing all the classes in the only in the master course, not for you people. Okay, at least I got 5 arts. Okay, expecting more. My heart will not be filled by just 5 arts. Okay, I hope like, oh, thank you so much. So a lot of people are, I think, see why I'm asking you this, like I'll be able to know like whether you are enjoying the session or not. If not, definitely we will try to improve yourself, and like there is always a scope for improvement. I agree with that. Even with the like 3 to 4 years of teaching experience, still the learning is always never ending process. So we will do it. We'll try to include the more interesting factors, interesting concepts, facts about the genetics, plus we'll stick our syllabus to the exam to exam basis, because the ultimate goal of you is to get the top rank in the exams, and we will give 100%, and I want you to give your 100%, so that later you don't have any regrets. Okay, so I'll be having one more session on the YouTube, so that will be in the tomorrow. So after that, we'll be continuing all our classes in our master course. So if you have not yet enrolled to our online master course, whether you are watching this video now, next year or next year, so for every year we'll conduct the master course for the ICAR PG for the 6 different subjects, 7 different subjects, including the plant science, and the complete course details will be available in our website www.agreedict.in, www.agreedict.in, or you can download the mobile application Agreedict in the Android or the iPhone. Plus, if you have if you need any additional assistance, you can call me directly on this number. Our student manager will answer all your queries and explain all the things in detail. Okay, so good night, everyone. I have not yet had my dinner. I'll have the dinner and sleep, and we'll meet tomorrow. And I am really very excited to join you all personally, privately in a live session through a Zoom, where you can open your like unmute yourself and interact. So that's where like the things get interesting. So I'm waiting for those things, those sessions that will be from day after tomorrow, and tomorrow we'll have one more session in the YouTube. I hope if there are any suggestions from your end related to the teaching, related to the concepts, related to the subject content, so you can definitely feel free to drop it directly in the comment sections. We will take it positively, and we will try to inculcate it in the upcoming classes. So all the class links will get in the Plant Science Master Group for all the topics. Yes, you will mostly get all the links, but students usually face problem joining the class through the Zoom link that is provided in the WhatsApp master group. So try to directly join opening the app or the website during the time of class. For example, if the classes are 10 p.m., just open the application by 10 p.m. and there you can get the symbol that live classes started. Click on it and you can directly join. Sometimes the students are facing the issues with the joining through the direct Zoom link. So better to avoid it. So can we get this class in the master batch? Yes, it will be available in the master batch only. The link will be provided there. You can directly watch it, and otherwise you can directly watch it in the YouTube too. Good night everyone. Okay, someone has said good night. Okay, okay, समझ गया. Good night everyone. I hope like you have enjoyed this session. So I am with a lot of energy and lot of positive intent to teach you all people in the smart board the very first time, since it is also very first time for me. So I'll try to practice more and more, and I'll come up with a more engaging content for all of you. Only thing from you, what I expect, the love and support that you have already provided us, keep providing it, and share this content with all the students who are preparing for this ICAR PG exam, PhD exam. Just let them know about the content, the quality of the content we provide, so that if they are interested to crack any exams, they can join us. Thank you so much. Okay, bye.